the report, so folks that are not with us, we can share it with them as well in our club. And Dr. Lips, thank you so much for joining us tonight at the HERP Club meeting. We're so excited that you're with us, and we can't wait to learn from you.
Well, thank you so much for the invitation to join the group. It's a real pleasure. I'm super sad that we're not physically face-to-face so we can meet and talk more, but I'm looking forward as the pandemic becomes less of a problem that we can eventually all get together and meet and talk more herps. I was asked to sort of give a talk on amphibians and the role of disease with the amphibians. And so I'm going to share my screen and see if this works. How's that looking? Good? Oh, all right. Thank you so much. So this talk is sort of an overview. I'm going to talk primarily about some of the work that we've done in Panama at the very beginning of the chytrid pandemic with BD, the original chytrid as it were, and talk about sort of all the different impacts it has had on amphibians, on the snakes, as well as on some new work we've got looking at the role on human health. And then wrap up a little bit, talk a little bit about the other emerging infectious disease of amphibians, the other chytrid, Bsal, which might eventually have some impact here in North America on our herps here. Knock on wood, it's still not here, but it'll be good to sort of keep an eye out for that. So, let me see, there we go. So I'm going to start off with sort of a real big picture with, amphibian biodiversity. I recognize that so many of you are already experts in herps. So this will be sort of a review for many of you. I think we all know that most biodiversity is in tropical regions, and for amphibians and reptiles as well, that totally holds up as well. And the reason I start here is because this sort of sets up this whole story of areas of the highest biodiversity also seem to be the areas that are most heavily impacted by all sorts of factors, but disease is one of them. So in this map, you can see the darker brown is where you have more species of amphibians. And of course, all the tropical areas are where you see the greatest number of species. As of today, it's not 83,000, it's 8,326 amphibians have been described as of today. I checked today. This number goes up every time I give this talk, and it's incredible, and it's important to remember that new species are being described all the time from all around the world, even here in North America, as probably many of you know. But this slide then is where we have globally threatened amphibians, and you can really see this same pattern then, where there's this red. It's the same places where you see the greatest number of species, right? So where there's the greatest number of species, we have really high threats to our amphibians as well. And so there's something about these places that are really important then, both in terms of discovery, but also in terms of conservation. And of course, we all love herps, and we know all the awesome ways in which they make the world a better place. But for most people, it's really a good reminder that, they play really important roles, right? They're especially important in regulating insects. So many of these insects are disease carriers for people. There's all sorts of incredible chemistry going on in their skin. People are discovering new biochemicals and biocompounds that hopefully might be of use to us in the future. And of course, they have some magic ability to regenerate, and there's a lot of work there trying to figure out, how we might use that as well. In terms of ecology, amphibians are super important because they are such a large amount of biomass. They're generally small individuals, but the sheer numbers of them are such that they play really important roles in moving energy, moving biomass between terrestrial habitats, aquatic habitats, from upstream to downstream, and then as they go through metamorphosis as well. So they're really important in moving energy and nutrients throughout an ecosystem. And because they're sort of right in the middle, it becomes really important for them to bring that energy to higher levels of the food web, as well as to affect the lower levels as well. And I'll come back to this sort of in the end there. So for most of us, the story began with the mysterious disappearance of amphibians back in the late 1980s. And at the time, I was a first-year grad student, and the last thing I wanted to do was get involved in something for which we had no idea what was going on. We didn't know where the problem was occurring. We didn't know when it was occurring. We didn't know what species were more affected. In fact, for about 10 years, people argued whether it was a real thing or not. You know, there was all this discussion as to whether these were natural population fluctuations. So it took a long time for people to really finally understand what was going on, because the reports were kind of coming in from everywhere in all sorts of different places. And it was really hard to know, is this one thing or is it multiple things? Well, as you are probably not too surprised, it turns out that it was in fact multiple things, but I'm going to talk primarily about the role of disease. And we'll see that the role of disease was especially important in some of what they called the mysterious or enigmatic declines. But it wasn't until 2004 that we really had an idea, any idea whatsoever, as to some sort of global assessment. And so there was hundreds of people who contributed to this report describing what was actually known about the status of amphibians. And so, for example, the recent work here in Maryland on the Amphibian and Reptile Atlas is sort of our current, most recent local version of this same thing, where you go out and you try and figure out, how are things going with all your species? What is the evidence that they are healthy or not? In 2004, the experts decided that amphibians were officially in the worst case scenario of any group of organisms, with 43% of species apparently declining, 32% officially listed as threatened or endangered, over 100 assumed to be extinct. And all this had happened really just in the past couple decades. So at that point, they could say that this was a recent problem, it was a global problem, and it was widespread. Oh, and here on the pictures, what I always like to do is point out that the 3 species there at the bottom are species that I worked with as a grad student that today are considered extinct. I have been in the unusual position of having seen dozens of species that simply do not exist today. So in very few years, this happened. And again, this is something that is not normal, it's not typical. And I put it up there really just to remind us of how unusual this situation is in the world. And that big assessment then highlighted these 6 causes of problems with amphibians around the world. And number one, as with almost all groups, is always land use change, right? We deforest, we chop down the trees, we drain swamps. All these things are bad for the amphibians. What I'm going to talk about are actually what we kind of consider novel threats. I'm going to talk about invasive species and emerging infectious diseases. And in this particular case, I'm going to talk about chytrid fungi, which are both an invasive species and an emerging infectious disease. And so it's quite similar in some sense to what we're seeing today with our own pandemic as another emerging infectious disease that is having quite similar impacts. So this would eventually be shown as BD, Batrachochytrium dendrobatidis, the chytrid fungus, the first one. And I put up this little cartoon really just to show us what is the life history of this little fungus. So here we have down here these little sperm-like zoospores. They swim around in a little bit of water. They look for a frog or a salamander, and they embed in the skin. And when those little zoospores embed in the skin, they drop their tail and they change into what is called a zoosporangium, these little round cells in the skin of the frog. And when those things mature, they have these little tubes that spit out new zoospores that then swim around and infect another frog. And so this just sort of shows the need for amphibians to either touch each other or to touch a surface or water that might be infected with the fungus. And that's how they get infected. The way the fungus kills the frog is by a combination of sort of two things. Primarily, the fungus fills up the skin with so many fungal spores, the poor amphibian can't get enough oxygen, can't exchange gases, can't get enough water across its skin. So if you look in the background of this slide, all those little round things, that's the skin of a frog. It's taken off of this guy here who's dying. And so if you peeled off his skin, you would see this layer of almost all fungal cells. There's a couple frog cells in there, but it's almost all fungus at this point. And so that blockage affects the blood chemistry, if you will, of the amphibian, and that then leads to all sorts of physiological problems, organ failure eventually, and the heart eventually gives out. And the way that this is able to happen is because somehow the fungus turns off parts of the amphibian immune system. Somehow it gets in there and is able to reduce the effectiveness of the immune system, which should be fighting this thing off. So the frogs recognize it as a problem. They start up their immune function to try and fight it off, but it's not enough. You know, they have not co-evolved with this fungus. And so they are still trying in that process of coming up with a way to fight off the fungus. Some species have certain ecologies or behaviors or genetic combinations that put them at less risk, but for the most part, most parts of at least the Americas, it's a brand new fungus and there's almost no resistance. So every species that has been tested can be infected. And there's certain combinations which allow the infection to either grow to a certain level that kills the frog or keeps the infection at a low level, and the frog or salamander can live with a low level infection. It's a little bit like COVID. If you get a terrible infection, it's bad news. But if you can keep it low, you can survive and potentially even recover. So this is a map. It's a little hard to see, but this shows us that just like COVID, there are different variants, genotypes, different genetic variants of the chytrid that are found in different parts of the world. This map shows the different colors represent the different variants. We see that green, which is called the global pandemic lineage, is everywhere. This is the one that is the primary cause of loss of amphibian diversity all over the place. But we also see purple, orange, and blue. And you'll notice that blue in particular is only found in Asia. So this is something that we sort of need to be aware of, because if we look at North America, yeah, we have the global pandemic lineage, but we don't have any of the other three. And we don't actually know what would happen if these other variants got to North America and infected our frogs, along with the one that's already here. So this is a good reason to prevent additional releases, imports of frogs from other parts of the world that might be carrying some of these other infections. And later on, we'll talk about the salamander chytrid. It's another one where it's not here. It's also in Asia and in Europe, and we want to keep that out especially as well. But this also reminds me that this is the source, as far as we know, somewhere in Asia. It's a little hard to work there, so we don't actually know the exact location, but we believe both Bsal and BD came from that part of the world, originated there and have then spread probably through human transport. So this is a figure from a paper where they basically sequenced the different types of chytrid in all these frogs that were in the trade. So they took these frogs from shipments, from pet stores, a variety of sources, and they were able to show in the different colors, the different lineages being shipped back and forth by us, by people, moving things from Asia into North America, into South America, huge transport hub here in Europe that just brings things in and sends them back out. They all get mixed. And this is terrible news, not just for chytrid, but obviously for other infectious diseases as well. So trade is really something that you need to be concerned about with the spread of infectious disease. And just to show one particular example, Elisa Schlegel many years ago swabbed a bunch of pet stores for chytrid, and basically 40% of all the frogs that she found had chytrid. And I think that says, it's hard for me to read here, 70% of pet stores that she tested had some infected animals. And that's because there is no legal requirement. This is not illegal. This is totally legal trade. There's no requirement for these people to do anything at all about infected animals coming in. They don't have to do anything. Obviously, they treat them when they're in the pet store because they want to be able to sell them. But this is a continual problem because these are always going to be an active source of infection and spread. And it's likely that these will be a place where we might detect new diseases that are coming in here. All right, so after all this time, a couple years ago, they got those experts back together and said, okay, now that we've had 20 or 30 years of work, what can we say has been the impact of chytrids on amphibian declines and disappearances around the world? So this is not all the causes, this is just chytrid. And the conclusion is that something like 500 amphibian species at a minimum, right? Because this is only the stuff for which we have good, solid data that have happened recently. Because all the old stuff, you know, there's not a lot of evidence that we can find that would indicate, A, what the population numbers were, when BD or other chytrids may have shown up, and then what the change was. So this is sort of tip of the iceberg type of numbers here, where it's sort of a terrible underestimate of what chytrid has done to amphibians globally. And you can see that where we have the most impact is in the Americas. And that's primarily because this is where it happened last. Once we were aware of the problem, we were looking for it, and people were out there counting frogs and surveying them. Places like North America, Europe, and Europe were probably affected a while ago. We don't know exactly when. There's evidence of declines from 40, 50 years ago. We don't actually know the cause. Chytrid was present. Did it make those changes? We don't know. Africa's just a black hole. We have almost no idea of exactly how bad the situation is because there's not enough actual field work going on in terms specifically related to population estimates. And you'll notice that Asia is not even on this map. There's no information coming out of Asia related to either population trends or patterns of infection for the most part. It's coming now, but historically there's been very little export of those types of data. So there's a lot more to be learned, both historically and currently, as to what's going on with chytrid and amphibians. But it's bad news, and it's probably an underestimate. But in all this time, what we've learned is that species do vary in their responses. You know, I mentioned a little bit earlier that a lot has to do with the intensity of infection. And so I like to use this figure to demonstrate how that kind of works. And anytime we're talking about disease, almost always, especially when we're talking about wildlife, we sort of imagine the outcome of disease is the result of three things interacting. The amphibians themselves, and there's a whole bunch of those. So each species varies in all sorts of different ways in ecology, in genetics, in behavior, habitat use, longevity, density. Each and every one of those things can affect its exposure to disease, its susceptibility to disease, and how well things grow in their skin. The pathogen itself matters, right? I showed you that there's all these different variants. There's probably way more diversity of chytrids than we understand today, unfortunately. They all vary slightly in terms of the particular temperature and moisture that they grow best at. Some are more lethal in certain species of amphibians than others. And then we have the shared environment, right? How cool or hot is the environment? That affects both the frog or the salamander, as well as the growth of the fungus, as does moisture and a variety of other environmental factors. So to understand why one species disappears while another one is able to persist, you sort of have to know a little bit about all these things and how they interact. Because ultimately, those species and those chytrids that are in particular environments that promote the growth of the disease, of the pathogen, and the frog cannot control it, those are going to be the species that are going to be most likely to die off. Populations will then decline and are at greatest risk of going extinct. And so there's a lot of basic biology that's needed here, as well as lots of lab work on the genetics and the immune system and all sorts of things. So lots of great work to be done. So most of my career I've spent in Costa Rica and Panama. And by pure random chance, I happened to be in the right place at the right time to actually watch this happen. I started out as a grad student, basically out in the mountains of Costa Rica, counting frogs, working on my dissertation, and happened to be at a place where this unknown disease at the time struck. But the story started a little bit earlier in Costa Rica. So again, here's Costa Rica on the left, moving into Panama here on the right. And let's see if this will work. And so the famous golden toad of Monteverde, which many of us probably know, disappeared around 1987 from northern Costa Rica up in the mountains here. I was working in southern Costa Rica, right on the Panama border, up in these mountains here. And basically I came back from a break at home, a semester at school, and the frogs were gone. And this is a remote protected area, hours away with four-wheel drive from any small town. There's nothing there. It's an international biosphere reserve. The next site was in Western Panama, just down the road. And I had moved there because I had literally run out of frogs here and I had to find a new site. And so I'd only been there a couple years and came back one Christmas and found dozens and dozens of dead frogs that were dying, salamanders that were dying. Just every day we went out, we would find more. And again, this was a protected park in the middle of nowhere, and it was unbelievable why they would all be dying. Everything seemed fine. There was no obvious problem, but it was the ability to collect those dead frogs and send them to a lab, which was critical, because that was really the very first time we had, sort of bodies to look at, send them off to a lab where you could actually do tests and see what they all had. And so it was those animals that were examined that were shown to have that thing in their skin that at the time was not described, that would eventually be discovered to be this Kytrid fungus. And then we could go back and looked at the animals from here, and it turned out that they also had Kytrid fungus prior to the disappearance later on. And so the rest of my life, I basically spent bringing my students down to these sites every summer, waiting for things to happen. We were sort of the advanced team. We ran thousands and thousands of transects day and night and counted and measured and tested for disease all the amphibians that were out there. And at the same time, we counted and measured salamanders, lizards, snakes, you name it. And eventually what we saw is that, this chytrid moved across Costa Rica and Panama. And we were a little bit lucky in that in this particular part of the world, it can only go one direction, right? If it is in fact a disease moving through naive populations, it's a narrow strip of land, and we happen to be able to track it by spending our summers living in the mountains here and waiting for things to happen. Most other parts of the world have been much more difficult because it could have moved out in any direction, and you would need many more people to detect that. But because of this, and because we were able to show it moving sort of north to sort of southeast westerly, it was allowed us to rule out a lot of other things. And then once we had the identification of the fungus in the skin, we knew what we were looking for. We could test things. And so today we have a whole set of tools and knowledge that we can use to be way more prepared for the next pandemic and hopefully prevent that from happening. But it allowed us to rule out climate change, which was one of the other sort of big ideas at the time, because we didn't see it going up and down the mountain. You know, it was sort of going east-west across the landscape. Not to say that temperature doesn't matter, but climate change was not driving the outbreaks here. It's sort of the main point here. So we actually watched it move all the way through Panama. By the time we got into Eastern Panama, we ran into sites that had already been impacted, because all of South America had been impacted decades before. It didn't get nearly as much press because it all happened before we knew what chytrid was or what was actually going on. And it's been sort of a retrospective historical study to sort of recreate the disaster that was chytrid entering especially the high Andes of South America. So at this point, chytrid is in all places and has really done most of its damage. But at this one site in central Panama, we waited basically for 12 years for this to happen, not really knowing when it was going to come, if it was ever going to come, what it was going to do. And so we were kind of forced to go back every single year and run all these surveys at this place called El Cope, right in the middle of central Panama. And today there's about 40 species of amphibians. There used to be at least 74, probably many more. And this is from one river valley. It's not a large part of the world. It's about 2 kilometers by 2 kilometers. And it had more species of amphibians than I think the entire state of Maryland. So you sort of get the sense of the type of loss that you're talking about here. So these are some of the pictures that we took, just a handful of, we found hundreds of dead frogs over this 5-month period. And you can see some of them, they clearly are not doing well. Some look fine, but they sort of stick their little butts in the air. And it's like they don't want to be touched. They're definitely not acting correctly. Animals in the middle of the stream that should not be in the stream at all. And basically what we were able to do with all that sort of dedicated field work is to show that over time, here's all of our transect data, is that, we waited and we got better and better at catching frogs. But as soon as we got our first positive test of chytrid here is when the die-off started. We immediately started finding dead animals day after day, populations just crashed. And they have not really ever recovered. Not all species disappeared, but almost none have returned to the levels that they used to be at. So this just shows some of the abundance shifts. If I just change the sort of the format of these data, we've continued some of these surveys. You can see here was the year right before BD arrived. Here's its arrival. And it immediately crashed down. So these are abundances, like density of frogs on a transect. And you can see it continued to go down all the way into 2010. And I had a student do another 4 years after that. And at that point, it kind of steadied out. It had not gone back up, had not continued going down. Chytrid is still there. It's still infecting most of the frogs. But the level of infection is very, very low now. So many frogs are infected, but for whatever reason, they have a low level of infection. And it could be something as simple as social distancing, essentially, where the density of frogs is so much lower that they don't run into each other. They don't run into more zoospores. And so they never really build up this huge intensity of infection. And so they are able to live with a low infection is sort of where we're at. Some individuals do get high levels, and so there probably is continued die-off, but it's at a low enough level, it's very hard for us to detect the dead animals. But let's look at what happened when it hit this site in terms of its effect on the tree of life of amphibians. So this is the tree of life of this location in central Panama called El Copay. And I worked with a guy named Andrew Crawford, who's a population geneticist and phylogeneticist. And really, you don't need to read all those names. It just sort of shows all the diversity of frogs. I've added a few pictures to give you an idea of like what we're talking about. The different letters represent different families. And red are the things that are gone completely. And you can see there's red all around the tree. So it's not just affecting the salamanders. It's not just affecting the toads. Any species anywhere along the tree can be heavily impacted and disappear. The names in purple are species that Andrew detected only through genetics. You know, we had been there 12 years. They all kind of looked the same to us. Turned out that genetically they were completely different species. So there's, again, way more diversity of amphibians that is completely unknown out there that really requires additional collaborations to even detect it, much less assess how it's doing. But this just shows that the evolutionary impact was huge, right? We have lost 30-some species completely, and many of the survivors are down to a fraction of their original levels. So the genetic diversity, no doubt, is also way down as well. And who knows what that means for evolution or survival or future generations. This is sort of the same data in a different way to sort of visualize, you know, what was sort of the responses of the different species. And I try and describe it as sort of these three general categories of response. We have those all the way over here on the right. These are the ones that are completely gone. So the golden toad disappeared completely. This gastrotheca completely disappeared. These tend to be endemic species, rare species, very specialized, cool, awesome species are the ones that are gone. The other extreme are common pond breeding, generalist species that occur over large areas. Things like the red-eyed tree frog didn't seem to be affected at all. Same with the marine toad. But again, both of those things, they move long distances and there's lots of populations elsewhere. And so they could easily have been recolonized from other places. Unlike these guys, which tend to be endemics to the top of the mountains. The third group then is everything in the middle. These are the ones that were reduced in abundance. They're not nearly as abundant as they used to be, but they persist. And, you know, they're sort of at much lower levels, but they're still there. They are susceptible, and many of them are still infected and probably showing some level of decline. But without intensive mark recapture, it's really hard to actually quantify the impact. I'm not going to spend too much time on this figure. What it is every bar is the sampling of the infected animals at a particular time. So for example, this first bar here is before BD. And so the amount of color in the bar is the percent of infected animals. Like if we went out and sampled for COVID, what percentage of the population was infected? The redder the color, the higher the intensity. And the higher the intensity of your infection, of course, means you're either dead, dying, or very, very sick. And so what this shows is it shows the epidemic curve, right? We heard a lot about it in the news. So nobody's infected here, BD appears, and immediately 80% of all the amphibians at this site are infected to some degree. And a whole bunch are dead, dying, or really, really sick. And over time, that number goes down, in part because there's hardly any frogs left by the time you get to 2010. But still in 2010, there's like 40% of individuals are still infected. And there's a bunch at this really high level. So it's not that the disease ever goes away. It is there. It's not nearly as bad because there's not nearly as many frogs for it to infect and build up really high levels in the environment. Because that's what happens with these chytrid is those zoospores are shed into the environment and somehow they're able to persist for some unknown period of time. So once many of the frogs are gone, if you put more frogs back out there, what happens is the remnant zoospores out there now have new hosts and allows the zoospore to replicate and increase and spread even more. So in this particular case, adding more frogs to the site allows potentially for future mini smaller epidemics, right? Because you're putting more food out there for the disease to feed on, replicate, multiply, and then spread out there. And so our normal conservation approach would be to do some captive breeding, put the frogs back out there, and everything would be great. But because we can't get rid of the fungus in the field, all of that is kind of not only not going to work, it's going to make things even worse potentially. So this has been a real struggle for us to try and figure out what do we want to do? You know, what do we ask the concerned citizen to do if our traditional approaches aren't going to work? And we still really don't have an answer for that. I'm not going to go through this. This really just shows that different species vary in terms of how bad things were. I'll just quickly highlight our Adelopus because that's the one that I mentioned that completely disappeared. And you can see that when BD first hit, something like 98% of all the Adelopus were infected and almost all of them had a really, really, really high infection intensity. And it's probably because that combination of really high intensity, really susceptible, and you're an endemic species that doesn't have any other populations out there is why it was able to be completely eliminated from this site. And so its abundance went to zero. These other guys, you know, they also were impacted. Again, everything was infected essentially, but the response varied quite a bit depending on how bad their infection was. Like these little glass frogs, they get infected, a lot of them get infected, but few of them reach that level at which they actually die. So they still persist at this site. So it's a really interesting story of variation in susceptibility, probably based on a whole variety of different factors that determine why some species have survived while others have not. So some of the ecosystem impacts have also been just as dramatic and widespread and terrible as the impacts on the amphibians themselves. We had a couple of grants that allowed us to collaborate with some stream biologists where we were looking at what happened to the stream, to the crabs, the shrimp, the fish, the tadpoles that live in these streams after the frogs and their tadpoles disappeared. And this has turned out to be a huge suite of impacts. I'm not going to describe almost any of them, but just simply say that everything we measured changed and changed quite a bit. I'll show you this one picture down here that tries to capture some of that. So here on the left, we have a normal stream before the frogs disappeared. And if you look closely on this mossy green rock underwater, you can see tadpoles attached to the side. These tadpoles are chewing away at the diatoms and the algae that live here. That has huge impacts on the mayflies and the stoneflies that live in the stream. It affects the amount of sediment in the water. It affects the quality of the sediment and the algae. And when the frogs died off because of chytrid, they didn't reproduce. And so there were no tadpoles in these streams for many years. And what happened is you saw the loss of the green, because this gross, slimy green algae started growing and it's not being eaten by tadpoles. And so it just takes over and it dies and it creates this gross dead layer that makes the rocks really slippery. And that prevents the stoneflies and the mayflies from getting to the diatoms. They go down. There's a lot of muck in the water now. It's not as clear. That increases the number of chironomids and other filter feeding insects. There's all sorts of changes in the aquatic insects that I won't go into. But it also had impacts on the snakes, and it's the snakes and the human impact that I want to sort of talk about next. Because of course, as a herp group, the snakes, those of you who've been to the tropics know that there are so many snakes in the tropics that are either sort of highly dependent or strongly connected to amphibians, especially as a food source. Here's just two that I showed here, but it's that diversity of amphibians and the high biomass, which is probably really allowed for the diversification and high biomass of all these really cool tropical snakes that do everything from eating adult frogs to specializing on arboreal egg masses on the leaves, to you name it. So, but as you guys know probably better than almost anybody, that snakes in general are tough to find, right? You are lucky to find maybe a couple in a day. In the tropics, that's true as well. And because you have so many more species, I think this particular site had something like, I think we're up to 150, 190 species just from this tiny little area. But almost all of them, we only have like 1 or 2 sightings in like 20 years. So this is the huge problem, is you can sort of see that there's changes, but to get enough samples, enough data to be able to statistically say that there have been changes is so hard. Because first of all, snakes are cryptic, they're hard to see, they're rare to begin with, like only a couple occur at high enough abundance that you see, enough to say anything in any particular year. So it took us almost 30 years to get enough data and come up with a whole new statistical analysis to be able to analyze the snake data. We knew something was going on early, but we didn't have the statistical tools to prove it. Because we expected, of course, that when the frogs went away, that all those frog specializing snakes would be affected. And this is an attempt to sort of show the change in the snake communities. And I'll show you some species-specific changes in the next slide. But essentially what we have here is in the blue is sort of the pre-decline. So we basically have 6 years of data on snakes along those surveys before the frogs disappeared, and 6 years of data collected on the same transects, collected in the same way after the frogs disappeared. And if you sort of create these little plots, this would be a representation of the snake community before, and here it is after in the orange. And what you see is that it's smaller, it's more compact after, meaning that the loss of sort of these unique elements has sort of allowed the snake community to become less diverse, both in terms of the numbers of species out there, but also in the kinds of species. Just like with the amphibians, what you're left with are primarily the widespread generalists, and you're losing the really cool, unique specialists on the frogs. And so the number of species that you see afterwards is much lower. It's on average 40 compared to the 52 that you saw before. And so the community is smaller in number, it's less abundant, and it's less diverse in terms of the types of diets and the different things that they do. Not all snakes responded in the same way. And this is part of the remaining mystery that has yet to be deciphered. What I've done here is kind of, here's our list of all the species for which we had enough data to analyze. And this over here is the pre-decline and then the post-decline numbers and how many detection events. And what I've done is I've added these lines in text. So these things up at the top that end up in the red, these are the species that were seen less often after the frogs disappeared, suggesting that they really depended on frogs to remain at some high level. And many of these things are things like Leptodira, which love to feed on frogs. But there's a bunch in here that don't seem particularly to be frog specialists. These guys at the bottom, where you end up in the blue zone, they actually increased in numbers after the frogs disappeared. And some of these guys, Bothrachus schlegeli, the eyelash viper you guys all know, but little dipsis, which feed on snails. So again, there's certain signals that food is part of this story, but there's species in here that you might not have thought that they would either go up or down following the frogs. And so there's other things going on that we do not yet know why they're responding in this particular way. If we just look at body condition of snakes, this was sort of how we knew something was going on at the moment, because many species, these four here in particular, we could see that they became emaciated after the decline. So a Cybon, Oxybelis, more Cybon, Ementodes. So before the decline, they were more robust, they were fatter and chubbier. And then after the decline, you could see that they were really in much poorer body condition. While these guys over here, they seem to be doing better after the decline. So again, there's some indication that this is a food story, but something else is going on that is going to require additional work. So that's the snake story. Really interesting, really tough to get the right data. And I want to end then with the human health story. These are some famous posters from long ago when the frog thing first came up, showing that, you know, we all sort of expected insects to change after all those frogs disappeared, because frogs are eating insects, right? And if you have that loss of so many species and so much biomass of frogs, you expect the insects to go up. And we did a lot with the aquatic insects. We did not actually do the terrestrial ones, which was a major problem for us, because of course, the thing you would most want to know is, well, how is it affecting mosquitoes or flies that might be transmitting human diseases? And so what we've been able to do is work with some modelers, economists actually, and they took our spread of BD through Costa Rica and Panama, basically the same data I showed you before. And we then were able to go to the public health ministry of both Costa Rica and Panama and see what the different changes were in diseases of people. And so here we see that in orange, we have Costa Rica and the number of malaria cases. And in blue is Panama. And you see that there's a shift. There's like a 10-year shift in time frame. And it turns out that 10-year shift is totally related to the timing of the frog declines in those two countries. So if you convert this really just to like how many years since the frogs disappeared did malaria cases go up, and combine the two countries, you get this down here. And so what you see is that here's 0. Here's, for any point on that map, the 0 point is frogs disappeared here. And then for 8 years following that decline, there's an elevated incidence of human malaria cases, suggesting to some degree that the loss of amphibians is somehow affecting malaria cases in people. And we suspect it's through changes in mosquito numbers. But those data, we don't have them, and it sort of sets this up then for the next big study. So we think this is really powerful, and we hope that once we can get this published, it's now a bioarchive and it's just been resubmitted. If it can be published, we suspect that there will be a lot more people like at the CDC or the WHO who might be willing to continue these investigations and help figure out exactly what is the role of amphibians in contributing to human health in the tropics and elsewhere. So there's a lot to be done there as well. But to kind of tie all this together then, you know, here's a picture of what we sort of see as the ecosystem, the food web of some of these different sites, with our frogs and our snakes here in the middle. We have our top predators. Here's all our stream system down here with the diatoms, the flies, the tadpoles. And we can show that the loss of amphibians from this central part in the middle has both bottom-up effects on the snakes and top-down effects on all the stream processes, but also on mosquitoes and human health. And so again, it took us almost 30 years to really pull all the different parts of the story together, because many of these things are not immediately obvious if you go out into the field. It really does require sort of this long-term dedication to collecting field data. hopefully will serve to encourage more people to do this and more funders to support this kind of important work. But the BD story then has been an example of how many impacts this one tiny little microscopic disease has on these ecosystems, the diversity of these ecosystems, and many other sort of cascading effects that are not always visible. I think we sort of are recognizing this with our own COVID pandemic now and sort of the repercussions this has on so many parts of our daily lives. But the amphibian story is quite similar in many of those respects. And so I'm going to end then with just a quick bit to highlight Bsal, the other chytrid that's out there that is not currently here in North America, and that we should be very, very concerned about, given what I just told you about BD. Because Bsal, probably many of you know something about it, is quite similar to BD. And even though we don't have a lot of field data yet and more lab data, we sort of are under the impression that whatever BD did, we can imagine Bsal is going to have the same effect because they're very similar in terms of their biology. So let me show you that and sort of see what we might expect to happen, right? So here is the map of salamander biodiversity. This is why we're all so terribly worried, right? So Bsal came out of here somewhere. The color is where salamanders occur in the world. And of course, the bright colors here indicate we have the world's global biodiversity hotspot of salamanders. We are in charge of protecting the salamanders. And given what we know about the difficulty in keeping BD out and controlling it in the field, it is terrifying to think that we are in charge of protecting salamanders, knowing that we have not been able to control BD, and we currently do not have any treatment for Bsal, right? So close up of North America and where our amphibians are, it's terrifying, honestly. We know that here in North America, there have been declines in many amphibians, but amazingly, I would say, other than sort of California, we know almost nothing about what really happened in central and eastern United States in terms of chytrid or these declines. There's lots of evidence of declines, there's lots of evidence of chytrid, but it's very difficult to link the two, as I'll show you. What we do know is that there seem to have been at least 2 waves of declines of amphibians in North America. The causes are not known. The first was sometime in the 50s and 60s. There was continued changes into the 70s and 80s in certain species in certain areas. And again, the cause, we don't really know, but if you go back through records for well-studied sites, there have been massive losses of amphibians. For me, one of the most incredible data sets is that of Dick Hyten, who is emeritus. He's still around at University of Maryland. He spent his life, 50 plus years, sampling plethodon from across all of Eastern North America. This is his data that we just simply plotted from his book chapter in the Lanu book. Every X is a place where the population declined, often by several orders of magnitude. And blue dots are places where there was no decline of those species. So these are a whole bunch of different plethodon species. You can see widespread loss of plethodon diversity across North America. Probably no actual extinctions, although the genetics of these things is quite mysterious still. But certainly we are losing abundance and probably some level of population genetic diversity. When I moved to Maryland and I saw this, I thought, well, here's my next research site. And I've spent a couple years on this and am hoping to get more money to follow up on it. We basically resurveyed as a starting point, resurveyed his transect across the Appalachians here. And we went back to his sites, resampled his sites in his manner, and tried to just recount what he had seen before. And what we saw is that there had been a shift. His early 3 decades, there was way more salamanders in many more places than in the past 4 decades. And this has sort of been a permanent sort of reduction, a shift downwards across all of that area. we thought for sure that we could go out and swab and we'd find evidence that BD and the museum samples that are all at the Smithsonian, that we would be able to show that, well, the declines happened when BD first appeared in those museum specimens. So we went and sampled the Smithsonian holdings. We went in the field and sampled a bunch more. And really, we can't find infected salamanders. You can infect them in the lab, they die in the lab. But almost everything that we have tested turns out to have no infection whatsoever. Now, if you bring them into the lab and turn the temperature down, all of a sudden some of them become positive and are infected, even if we don't give them disease. So there's something about temperature in some of these animals that may be part of the story. The fact that they live underground and it's very hard to see them makes it very hard to know what's going on. But we know that chytrid is present all across North America. This is a map of just chytrid presence absence from places that were studied. And you see that's everywhere in North America, especially here in the East. So certainly the conditions are right. We have the species, we have the fungus, we have the temperatures. But again, that connection between the presence of disease of the chytrid and population declines is difficult to make. In part because there's not a lot of historic population data. There's a lot of presence, absence, and collections, but less on the populations. We know from USGS Army work that there are currently annual declines in amphibians all across North America. They have a bunch of species that they've been monitoring for many years. And many salamander species, especially across the southern part of the US, are declining. On average, it's something like 3% declines per year in all these things. So there are current declines happening. And again, these guys also look for chytrid and, BD is out there, but they can't actually say that that is the cause of these declines. But they also can't point their finger at anything else either. So it's really disturbing here in North America with incredible resources and tons of herpetologists and people in the field. We still have a long way to go to understand what is going on with our own salamander diversity and populations. And of course, this is all pre-Bsal, right? So let's take a quick look at this Bsal Kitrid. It's a bit of a misnomer now. At this point, we know that it can infect frogs and toads as well as salamanders. So this is terrible, terrible news. I'm going to show you a couple pictures. This is what sort of BD looks like at the top. It's more of like a carpet, and then the skin lifts off, and it's like a lawn of chytrid in the skin. Bsal is not like that. Here's a picture of a newt. I'll give you a close-up in a second. They're actually more like lesions or ulcers. And so if you see, and newts in particular are more affected than any other species out there so far, all the different types of newts. So if you see a newt and it's got weird lesions, that would be something to really pay attention to. So instead of this carpet, they have holes in their snout. And instead of like the whole belly or the top side, it tends to be in little corners and nooks and crannies. The eyebrow, behind the arms, the snout, you can see the little holes here. Those are all Bsal lesions. This is an animal in the lab. So these are the things to look for out in the field. If you happen to see these, please contact somebody for follow-up. Here's the life cycle of Bsal. We have toads that now can be infected. Here's our fire salamander, the one thing that's really shown declines in Europe. We got newts that are infected as well. Here's our zoospore, just like BD. And here's the other really bad news here. These are encysted fungal spores. This does not occur in BD as far as we know. These are little floating sticky spores that when the duck or bugs touch them, it sticks to them, and then the bird flies away and brings it to the next pond. So if anything, Bsal is kind of even more likely to be spread because it has not just this, but it has this form as well. And again, don't think it's just salamanders. Frogs and toads can also spread it. So this is something, again, to keep an eye out for, and any sort of dead or dying animals will be highly important. But again, you know, with this, just like with BD, it seems to suppress the immune system. The intensity of infection seems to be a key factor. It causes massive mortality, really high levels of death. I've shown you the different additional levels of transmission and spread. Again, bad news for North American salamanders. And this is old news from Vance Friedenberg's lab, where they kind of indicated here's where Bsal would be most likely to live based on temperature and moisture in the red and the orange and the yellow. Here's where salamanders are, of course. And then here in the bottom is the combined map. And so the southeast is going to be ground zero for Bsal, probably. And those numbers are just, those are the main import airports for amphibians. So we can see that this is sort of where we want to keep our eye open in particular, and not being too far away. We kind of have an important role to play here. So is Bsal the next pandemic? We hope not, but again, we don't have any treatment. We need to keep it out. It is in the pet trade, of course. It has already been introduced to Europe. And again, knowing that it came out of Asia, they can see it in historic collections for at least like 150 years. And they don't know how it got out other than through our trade of pets primarily. So to wrap up, you know, the Bsal story, we're still learning about it, but it seems to be quite similar to the BD story. If that is the case, we need to be very careful because there's no cure for wild populations. We need to do everything we can to keep these things out as long as possible in the hope that somebody comes up with some sort of miracle cure here. And they all persist in the environment. And this is just the worst possible combination where, you know, if it gets here, we can't get it out of these sites. And then how are we going to help recover these populations? So again, I think more than ever, we are all so aware of the importance of emerging infectious diseases, how these things are spread globally like wildfire, and how important it is to keep our eye out because, of course, it's not just wildlife, right? We ourselves now are going through this very same thing. And so hopefully we can use this as a way to communicate to people in charge who can perhaps help us prevent the next pandemic going on there. So thank you all so much for listening. I know it went a little bit long. But hopefully somebody has a few questions and I look forward to talking to you some more. Thank you.
Wow, Dr. Lips, that was amazing. Thank you so much for sharing that. I mean, it was amazingly disturbing in so many ways as well. Any questions? Just raise your hand and you can unmute. Joe has a question.
It is often said that changes in the ecotourism or visitations to sites by researchers or other, and I'll call them naturalists, may go ahead and spread the particular diseases that we're finding in animals. Does your study has it ever pointed to the fact where you've noticed that there's been an increase in ecotourism in an area or research in an area that may indicate that it is people who are bringing the particular fungus to other particular areas and starting the process all over again?
Yeah, it's a great question. We have no evidence that shows that. It's certainly possible, but I always like to say that, you know, certainly we all do, you know, the sterilization of our boots and the cars and the tires and the nets and all that stuff. But there's tons of people, as well as wildlife, cattle, horses, birds, lots of other things that move around as well. So it would be difficult to attribute it to tourists or human visitors and not be able to rule out pretty much anything else. At least at the sites that I work at, like one of our sites, you've got water buffalo and cattle and people walking through the park all day long, every single day. So could be anybody. But we know from how it's moving, you know, sort of this, at least in Costa Rica and Panama, where we have good data, right? These are not along trails. Like the trails and the roads go from the coast up into the mountains and back down. It doesn't go across the top of the mountains. And so it is able to somehow get along on its own by itself. So we can't completely rule it out, but it's certainly not the only thing either.
Are there any other questions? Karen, it seemed like with the up and down oscillation of it was almost like a predator-prey relationship. So that when it goes away, then your predator's going to go away, and when your prey comes back, then the predator is going to come back. It just kind of seemed that way to me when I was listening to you.
Good eyes, that is exactly what happens, and that's often how we describe it, right? So everything is great, and then they crash, and then there's not enough resource for the fungus, so the fungus becomes rarer. That allows the frogs to come back a little bit, but then the fungus comes back, so now it's got more to feed on. So they never quite get back to where they were, and so it's these dampening oscillations where every time it goes down a little bit more, it can't come back up where it was. So yeah, you're totally right.
Eugene has his hand up.
Yes.
So really a wonderful perspective in your talk. What do you see as good protocols for sterilizing your boots in sites you visit in the United States now?
Yeah, so the official protocol is something like 10% bleach solution. Like, you know, if you can get most of the mud and gunk off your boots, you're probably doing a good job. If you can then soak it in, you know, low percent bleach solution, that will for sure kill it. You know, the reality is that we don't, at this point, the one thing that we have is kind of everywhere, but, you know, the concern is that maybe near cities or airports or import places, that would be places where there might be new introductions. So a bleach solution, there's some different sprays you can get to bleach your boots and your tires on your vehicle if you wish. Certainly the nets are something that we pay a special attention to. We usually leave a net and we don't move it at all. So
one of the sites that, or two of the sites that I visited a lot in the past get occasionally flooded by rivers. And what do you think of the idea that at those sites, that no matter how much you clean your boots, the main sources of infection are really beyond your control? What do you think of that?
I think about that a lot because, you know, I'm one of the people that they accuse and they say, well, you know, the herpetologists are out there and they go there and then everything dies. And I'm like, well, we are in fact bleaching our boots. And so as somebody who has heard these words before, I will tell you that being able to say that we clean and sterilize all our gear all the time gives you that sense of, okay, I've done everything I can. We can't see these things. That's the problem, right? If we could see that, if they glowed or look bright red, then we would be able to be 100% sure that anything that we're doing is good. But because you can't see it, you don't know. And just like with COVID, you wear the mask. And it's sort of like bleaching your boots is wearing the mask. So you're sort of doing it so that you have peace of mind and know that you've done everything you can to not infect others.
Any other questions? Raise your hand or make a note. Christina has a question.
Okay, can you hear me? I was wondering if there was a push to either work with researchers in Africa and Asia or a funding push to try to direct funds there to help kind of get some more information about what's going on there and how land use change is really sort of impacting that and providing more possible opportunities for these infections to spread.
Karen, you're muted. Okay.
Yeah, sorry, somehow that got clicked over. Yes, that's a great question. I think that, you know, Asia is sort of its own particular case because much of it is actually China, North Korea, where, you know, there's not going to be a lot of foreigners going in and explaining what's going on there. So you sort of rely on in-country experts, and, you know, it's this type of thing is not, I think, high priority, especially at the moment. So it's a little hard to know what's going on in Asia. Other parts of Asia, you know, the southern part like Hong Kong and Vietnam, there's more and more research being done there all the time, and we're getting better and better data. It tends to mostly be taxonomic and sort of people go out and survey for disease, but to actually do the marker recapture that you actually study populations, this is not something that is, well, it takes a lot of time, it's a lot of work. And so it's easier just to go out and collect and you're done. Rather than live out in those remote places. So there's more information coming in from Southern Asia for sure. Africa is another tough case because, you know, you've got other security issues there where you can't go out at night in many places because of the predators, you know, the lions and the leopards, and there's a lot more diseases and hippos and crocs in the water. So it's like you have a lot more sort of security risks for the surveyors. I know that there's a variety of organizations trying to encourage more research on certainly the amphibians, but you know, the problem has been is that it was like a hot topic for a while and there was lots of organizations supporting it. And now of course, everything is COVID and bats and viruses. We kind of try and make this the case that, well, don't forget about the frogs and the fungus, because this is really just, again, yet another case of these emerging infectious diseases. And work in Africa is desperately underfunded. And the problem now is it's just these types of things are no longer top priority. I mean, infectious diseases are, but of wildlife, if you can't connect it to human health, it's a lot harder to get that type of buy-in, I think, but it's totally needed. Totally agree. Eugene, do you have another question?
Yeah, the malaria connection is the first one that I've seen that would actually get lawmakers to open the taxpayer purse. And so getting that into print and then getting good publicity on that will be important. I worked for quite a while on deer impacts and the extraordinary impacts that deer are having on our forests and on our wildflowers, that to the extent that our forests are really changing before our eyes. And guy who was head of a big regional conservation effort gave me a strong lecture and said that of all the things that I talked about of the deer impacts, that people wouldn't care until I mentioned human diseases, which of course would be Lyme and ehrlichiosis and the other tick-borne diseases, and a second, deer vehicle collisions. So, and you probably thought a lot about this yourself, that the malaria connection, that might have, just throw out a number, 100 times as much impact as the loss of snake diversity and or the changes in stream flora.
Right. That
people in the herpetology group would say, that's really important stuff. So
yes, you're totally right. And we've gone through this. I remember one particular conversation many years ago where I said something to the effect of, yeah, it's affecting the snakes too. And somebody said, well, if people don't care about the frogs, you think they're going to care about the snakes? No way. You know, snakes to most people are not something to protect. So I totally agree. And if anything, we look at that malaria paper as the way to really push for greater conservation of wildlife in general. Because if frogs can affect mosquitoes and malaria, then the birds, the bats, you know, all the other insectivores can as well. And it's just one example that shows you the broader importance. And it's a little bit similar with the pandemic, right? Because if we can sort of build off of the interest and the concern of the COVID pandemic and say, well, there's the same thing going on with wildlife and it's having devastating effects there. And then say, and losing the frogs and the bats and the birds is going to affect Lyme disease, malaria, Zika, all these things. Then it gives you that extra push to do something about it beyond, yes, these are beautiful, awesome organisms. So we're trying. Yeah.
Yeah. And I've also looked into the history of demonstrations and public movements working for change in a variety of things since the 1960s. And one of the long-term messages is that people who want very specific goals that are attainable, that those get to be done. Whereas people who have vague goals, where we think they know what it means, but you can't really define it, those things hardly change. So with the pet trade, if one of your goals, and I'm just trying to echo if I understand what you were saying, is to stop all imports of a particular tax. So let's say salamanders and frogs from the rest of the world, and to look at this in a variety of ways, then that specific goal could be met. Because
we meant it a little bit because, and so this is where we sort of are working, because as you say, like there's lots of things that matter, but in terms of telling Congress or Fish and Wildlife, here's something you can actually do that will have some particular impact. We as a community have been giving Fish and Wildlife all the data as it comes in on BD and Bsal, and they have been very concerned about this, and they're doing everything they can, but they can only do so much under the legal authority that they have. And so they did put in a rule to block the import of 200 species of salamanders. And now that we have a new administration that's more interested in science, the hope is that they will revisit that rule and add other amphibians or provide testing or something that would at least tell us what is coming in and prevent the import of the infected ones. So people are working on that. I totally agree. I think you're right on the right track. And if people have other ideas for things that could be done, both in terms of sort of the policy angle or research, please stay in touch and drop me an email, because if people have ideas how we might combat these chytrids, I'd love to hear it, because we've been trying to come up with solutions for decades, and it hasn't been easy, and it's not really clear, because I get to meet all these great people at meetings like this, and people are concerned and they want to help, but I can't tell them what to do other than the usual, donate to places where they protect and conserve land, and do all the good things with, not spraying chemicals and recycling. But in terms of specifically helping amphibians, there's fewer options there.
Dr. Lips, has there been any surveillance or look at, I know a lot of the Asian food markets locally sell live bullfrogs, which are a big carrier for BD and Bsal as well. Is that correct?
I don't know if they've shown that bullfrogs carry Bsal, but other frogs can, but I don't know if they've been tested yet.
And a lot of those bullfrogs are actually imported from farms overseas, and then even folks in our own group may well-meaning want to save these frogs from these food markets, and they're kept in really high densities and really unsanitary conditions. So a definite disease reservoir is there. And it's pretty common. Not a lot of people know about it, but yeah, these bullfrogs are in these markets, and it's definitely a source for it.
Well, the markets, yeah, you're right. But even pet stores, like we've heard stories, there's like a couple of these unpublished things from years back where somebody did a survey of like elementary school teachers, And of course, they all want to have like a pet frog in their classroom that they get often from a pet store. And oftentimes the frog gets sick or dies, or looks to be healthy, but winter comes and the kids no longer want to take care of the frog. They don't want to kill it. And so they let it go in the pond nearby or in somebody's backyard. And this is just something that people don't want to kill the frog, but letting it go might actually be worse for many other frogs. And so something like that is, I think, a message that the average person doesn't maybe think about and might not be totally aware of, these mysterious invisible diseases that our pets might be carrying. So that's another angle that could go a long way. Especially you guys, where you're going to interact with so many school kids and teachers.
And on that line, did Schlegel ever redo the pet store study again, just to see if that trend has changed any?
No, I don't know that she's doing that research anymore. There might be other people doing studies, but of course the problem now is that the pet trade is a little sensitive about this. They're all private. You can't actually we've tried, getting some of their information for a variety of reasons, and you can't, it's not publicly available. And unless they cooperate, you can't tell. And so they had a big effort at some point. They called, I forget what it was called, like Save the Fibs or something like that. And they claimed they had this chemical that they were treating all the surfaces in their rooms and containers, but there was no way to verify that. I mean, they may have been doing it, but there was no certification or anything like that. So part of this has to sort of really eventually get to, what is the pet trade, and can we stop taking animals from the wild and find some way of, some sort of certification program for captive bred animals, because I think we all know that there's lots of claims, but there's almost no way to tell where animals actually come from. And the reality is, I think much of them come from the wild. And not only is that bad for the wild populations, but those are going to be the things most likely to be infected with any number of diseases. So.
And in your talk, you are encouraging us, if we find any amphibians that look impaired, to take note of that and let you know.
I would certainly, I mean, you guys, of anybody, right? You are experts. You know what these animals are like normally. You would recognize them when they're acting weird or sick, or you find dead things. And it sounds like many of you work with Scott from DNR. So, anybody who sees anything suspicious, I would say get some photos, get a GPS locality, probably can't collect without a permit, but get the information to somebody who could at least go and check it out. And there's a bunch of people in this area with labs that we could quickly tell you, what's going on. And better safe than sorry. If it's nothing, no big deal. But, if it's something bad, we want to know as soon as possible. And a couple of years ago, USGS had a meeting, and there's actually like this, we sort of demanded that they create this, they wanted to do this management strategy. And we're like, we can do that, but we want like the emergency response protocol. Like, who are you going to call if you find a dead salamander, right? What do you do? What does the average person do? Who do you call? And I think you guys kind of know, you probably have a whole list of people you could call and say, listen, I don't know what's going on, but here's some information. Let's go check it out, because it's people who are in the field who are going to notice it and can get the word out to the right people quickly.
Well, this has been wonderful, Karen. Thank you so much for sharing your knowledge and expertise with us, and for actually giving us some ways that we can participate and help, I think. So that's
absolutely
great. And I hope this isn't our first, our last time working with you, now that you know where we are. And I hope that you'll get to come and visit in person too next time.
Yes, please send me the info on your BioBlitz. I'll see if I can get out there. I do hope to meet you all guys in person, and might show up at one of your upcoming in-person meetings later in the year. And again, please stay in touch. Please stay in contact. If there's anything I can do to assist you guys, please let me know. But it's groups like this which are like the heart and soul of conservation. So thank you all so much for the invitation and also for your service to the amphibians and the reptiles.
Thank you.